And they get stuck, and then if they've got
Yes.
teeth, just let go.
That is true.
Oh my god, like almost a 3-pound catfish, and then it bit my daddy from it. Biting. They don't even have teeth. They just have like that really sharp, bony plate on top of their mouth. But still, yeah, it's biting.
Very good.
Bite the wiggle.
All right.
That's basically the best they tell you when it's bite and wiggle.
All right. I released the grades for part 2. So they're on Blackboard, and I will bring the part 1 exam to lab. So we'll have those, and you can see them. Next week, in lab, after we do our lecture part, we'll have not necessarily a retake, but we'll have a second attempt at the closed book portion. That'll be up on Blackboard, so probably want to bring some scratch paper or grab some out of our recycle bin over here, and a calculator. You can also bring your t-tables and normal tables, and I'll bring the stack that I printed out, so we have it. All right. Yeah, I do. That I didn't post it yet, so yeah, I'll get it posted. I was gonna stick it on the bulletin board, but yeah, I'll get that posted. Yeah, application part was okay. I guess we'll talk a little bit when we get the handouts, or when we pass back the full exam. Yeah, those grades were okay. They were better than the closed book, which was kind of expected. Usually is. But, all right. So we're doing confidence intervals. We're doing bootstrap confidence intervals. So we can set up this whole bootstrapping procedure where we shuffle our vector, calculate the mean of that. I shouldn't say shuffle. I should say sample with replacement our vector to get a new vector. Calculate our test statistic from that vector. All right, repeat it over and over and over again until we have a collection of these randomized test statistics. And then when we calculate the 2.5% and the 97.5% percentile, all right, that gives us a 95% bootstrapped confidence interval. All right, so I asked you to kind of figure out the fish, do one for our fish. So our problem said we had 50 fish and we had 10 were infected. So what we could do is create a vector of zeros and ones, where 0 represent the number of fish that were uninfected, and 1 is the number of fish that were infected. And we do that because once we create our vector and we calculate the mean of that vector, it tells us the proportion of ones in our data set. So it's really useful to recode these things into a binary format, zeros and ones. Now what we want to do is create our confidence interval. So our bootstrapped CI, all right, we've got our chunk. So we go back up to our generic function here, the generic chunk. I'm going to go through, copy that, move it down here, paste it, and the one value that we have to change is this, data $vector. Now, we don't have a data set attached to it. We created our own vector, and the vector that we called it was fish. So that's all that we have to use, is take it out and get fish in there. And once we have that, just go ahead and run that chunk, and I get a confidence interval of 0.1 to 0.32. So our 95% bootstrap confidence interval, let's do like a write-up. In our collection, 20% of fish, and we would do 95% bootstrapped CI, would be 10%, 10 to 32%. In our collection, comma, 20% of fish, were infected by a parasite. So we did our write-up where we give our proportion, which would be our mean in that case, and we give a confidence interval. Now what you see is that if 20% was our mean, 20% of fish were infected, our confidence interval went 10 percentage points below that, but then 12 percentage points above that. Are we symmetric around our mean? No. No, we're not. We are asymmetric. And that's a nice thing about these confidence intervals, these bootstrap confidence intervals, is that we're not making assumptions that we follow a normal distribution, and thus we don't necessarily have to have a symmetric symmetric confidence interval. Our confidence intervals will reflect the underlying data. So when we deal with these binomial type of data, yeah, we're symmetric when we're right around 50%, but as we start to get closer to the extremes, we should start to see asymmetric error bars, and that's what this reflects. That's what that reflects. So, right, properties, we don't assume any sort of underlying distribution, and that's different than when we do a regular confidence interval, what we would call a parametric confidence interval, and use the t-distribution or the qt function. That assumes a normal distribution. That assumes normality of our data. We don't make that assumption. We can calculate standard error and confidence interval for nearly all statistics. We just demonstrated it here with our fish example. The fish example is just a proportion. It's a single number. And you could say, well, how did I get this confidence interval when it's just a single number? I didn't take multiple samples. We had one sample that gave a single value, 20%. Well, bootstrapping allowed us to generate these intervals. We can't do that otherwise. Confidence intervals reflect the underlying distribution. If we're skewed, our bootstrap confidence intervals will be skewed. And it'll be skewed in the direction of that heavy tail. And then lastly, our bootstrap means will be centered on the sample mean. So I can go back just to kind of demonstrate this. So our mean was 20%. That's what we had. So if I just calculate the mean of our mean, which was the vector that we used, you're going to see, and I'll just bring this up, you're going to see that we're going to be close to our actual mean of our data set. And that applies universally. That applies universally. So there's a lot of pretty cool aspects to it. All right, so here's one more practice. All right, so we're going to generate random data, and we're going to save it as dat.x. I do have set.seed in here, so that way we can get our same vector. Yep.
So you started the 2D discuss section at
Yep.
the end of last class.
Yep.
Are we getting to that? Or?
Yeah, we will. We're going to put our practice before that to be discussed.
That's what I wanted to make sure.
Yep. Yep. So as I do have the to be discussed here. So before that, I'm going to do a practice problem. So we're going to generate our data where we set the seed to 2015, and then we're going to run this, and note where I put it. I put it above this to be discussed. We're going to do a chi-square, so chi-square distribution is skewed. I want you to generate this dat.x vector and then calculate the standard error and confidence interval for the dat.x. And I will do the same thing. All right, how's it going? You get it? Hopefully it didn't take too long. All right, so I made my vector dat.x. If we were to look at it real quick, I can do hist of dat.x just to take a look at it. And let's do, so you can see the histogram is skewed, right? I mean, degrees of freedom, 4 degrees of freedom, really easy. We could add a curve to it if we wanted to follow the chi-square. You get the idea. We're skewed, all right? But that's not our focus. Our focus is, let's see here. There it is, that's better. All right, so our focus, I look at the mean and standard error. This is what we have for just a regular vector. All right, we would report our mean. We could report our standard error, but we're skewed. All right, we're heavy in our tails. So a better measure of dispersion might be these bootstrapped confidence intervals. So bootstrapping, I just copied and pasted that chunk with the bootstrapping. I replaced dat.vector with dat.x because it's just a vector. We don't have it in a data frame. And then when I run it, I get this range, 3.00 to 3.88. All right, so our bootstrap confidence interval of the mean is 3 up to 3.88. All right, what about the standard error? So when we have a bootstrapped data set, the standard deviation of that bootstrapped data set is the standard error of the mean. So that's just kind of a definition, something to know. And you'll see that our standard error, bootstrap standard error of the mean, or the standard deviation of that, is very close to the actual standard error of our vector. So, you know, again, that's another pretty decent property. We're gonna be close. So these two values are close. So I did that here just to kind of make sure you have it in the note. For bootstrapped vectors, the SE of the mean is the standard deviation of the randomized we'll just keep the same term, bootstrapped vector. So the standard error is the standard deviation of that vector. All right. Make sure you have that in there. To be discussed. What happens if we have missing data? What do we do? So let's generate a vector of missing data. All right. For example, we will do this. We'll do M data for missing data. All right. And what we will do is we're going to make a vector, and we're going to use let's just make some numbers. We'll do 8, 6, 7, I'll do an NA, 10, 8, 8, NA, 6, 7, 10. Just some random data, where the NA is our missing data. All right, hopefully if I run this, I don't get a mistake. Yep, so when I type in M data, do it, we have missing data. 2 values that are missing. All right. If we calculate the mean, and standard error of this vector, we can do it. We remove NAs and run the calculation. Right? So for us, If we do the mean, we could do mean of that m data and then na.rm equals t. And we already built in the na.omit function in our standard error function. So we get these values. We get those values. They work. But what do we do with the bootstrapping? What do we do with our bootstrapping? So how should we perform bootstrapping on a vector with missing data? Option 1 is to remove the NAs, and then do the bootstrapping procedure. All right, so for example, our option 1 could be mdata, mdata.save, right? na.omit of mdata. So we strip out all of our missing data, and then what we can do is use our function. So I copied and pasted our bootstrapping and run it this way. So first strip out the NAs, and then bootstrap all that's left. That's option 1. What do you think? Does it work? It works, right? I get answers. What do you think about this? Bless you. What do you think of this? Do you like it? You don't like it? na.omit. We stripped out our missing data. Okay. 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 Yeah, so right now we don't have anything in here that says na.rm equals t. Right? So if we had missing data, this is gonna fail, believe it or not. That's going to fail because we have missing data. And we could sue it. I could do, let's see here, mdata. You'll get this. Missing values and NANs not allowed if na.rm equals f. Right? Error and quantile, it's just all, it's all messed up. You'll get that. So, do you think it makes sense? Right? Data aren't there, we take it out. Okay. What about other people? What about option number 2? And bootstrap the vector. This requires adding na.rm equals t to our code. Consequences of this are, or the consequences are that we can get more NAs than what was observed. So I'm going to paste that. There's our vector. I'm going to do mdata because that's our data set. But now what we need is this at the mean. So na.rm equals t at that point. So we add na.rm equals t. So that way, we're going to bootstrap here. We're going to sample with replacement our vector that has the NAs. So it's entirely possible that a new vector is missing all of the NAs, in which case the mean will work. Or we'll have 1 NA or 2 NAs or 3 NAs, or maybe even 4 or 5 NAs in our data set. But we're allowing the possibility of getting NAs, actually drawing NAs for some of our sample. So now if I calculate this, it works and I get a confidence interval again. What do you like it? What do you think about this one? You like this one better? Why?
Actually, I like it because if part of your experiment is the fact that sometimes you just don't get data for it,
Okay.
then it makes more sense.
Yeah, so
Like, I
part.
think it depends on if you want it, if it's important to include if data is missing.
Okay, that's kind of the key. Is it important to include if data is missing? So here's an example. We have a student who goes out and looks at nests of kingfishers. All right, and when he finds a nest, he takes measurements. How high is it above the water? You know, what type of vegetation is overhead? Or maybe, you know, how close it is to, you know, a tree or something like that. And some of those nests have trees close by, others don't. So instead of reporting, you know, infinite distance to the closest tree, it's just missing. In that case, I think it's probably important that sometimes the data is missing. So I think you could then argue that we would include the possibility of actually drawing missing data with our bootstrapping. But if we were doing something like canine lengths, right, on skulls, you did a right canine, you're measuring it, some of those skulls are missing their canine. All right, but you took other measurements on the skull, so that missing data is actually in your data set. Would I include those NAs there? No, I think I would argue for option 1 because we know that they were likely born with 2 canines. It's just they lost it at some point. So if we're looking at representing averages and dispersion for that value, then maybe option 1 works. So there's 2 options, and this is just kind of gets back to, you know, what's our philosophy? What does the data look like, and how does that direct us to whether we include the NAs or we drop the NAs? Is there a right answer? No, probably not. Probably not. But I would say make your decision quickly and just run the test. All right? Because you can't really be wrong.
The 97.5% confidence intervals are different. So
How close?
Yeah, it's not far off, but I was going to ask which one's more accurate. But I guess.
Oh yeah, these. Well, so it's probably they're both accurate because we're both going to be centered on the true spread. I think we're going to be a little less precise because you have the possibility of having NAs. So we had, I don't know, what was our length of this data set? 11.
Wouldn't your precision though depend on that like whether or not the missing data itself is data?
Well, yeah, because it changes our sample size.
Yeah.
So we have 11 data points in our M data. 2 of them are missing. So when we resample, there's some cases we'll have 11 data points that allow us to get a mean. Other times we may only have 5 or 6 data points, just because of the way it happens. So the fewer the data points, the less precision we have. So that's kind of where I go with the precision. And the precision here, you know, so accuracy is how close our sample mean is to the actual true mean, the mu. Our precision would refer to our variance of standard deviation. Less precision, larger confidence intervals, larger standard deviations, larger standard errors. All right. Yeah, it's which one's better? Again, just make a choice. Don't sit there and spend 2 weeks contemplating should I or shouldn't I include it. Just pick one and run with it. Hopefully that kind of saves you some of the headache. All right. So power of a test. We talked about type 1, type 2 errors. We talked about alpha and beta. Power of the test is the probability of rejecting that null hypothesis when it should be rejected. All right? And we always strive to pick the test, pick the procedure that has the highest power, because we want to be able to reject our hypothesis when we actually need to reject it. So I just kind of put together this table. We've kind of seen it before in terms of the Bayesian statistics. The actual, our null hypothesis is true or it's false. If it's true, we want to have a situation where we fail to reject it. So you conditionally accept. And we don't really have any good term here other than like 1 minus alpha. That's it. But if H is true and we falsely reject it, that's our alpha. That's our type 1 error that we're looking at. If H is false, but we fail to reject it, that's our beta. That's our type 2 error. What we're going to look at is this power, because that's what we want to try to maximize. All right? We want to always try to maximize the power. So what are some generalities? What are some rules that we can go by? One, the test, the type of test that we run. We're going to learn how to do parametric tests. That's like your t-test, your ANOVAs. They're called parametrics because we make assumptions about the underlying data. Specifically, we assume that they're normally distributed. All right? Whenever we do that, the power of our parametric test is going to exceed the power of a non-parametric test. What's a non-parametric test? It's an alternative way of accounting for some of our violations of the assumptions. Usually with our non-parametric test, we take our data, our actual measurement data, and reduce it in some ways. So we throw out some information from the data and run our tests on that simplified data. Thus, our power is going to suffer because of that. All right. Note, though, that if we're running a non-parametric test and we still detect significance, so we reject our null, then we will always reject our hypothesis for a parametric test. So it's one of those things where our data, they meet all the assumptions of the parametric test, but you say, well, I want to run this non-parametric version. If you get significance, no problem. That's great. Because if you have significance for the non-parametric test, you'll always hit it for the parametric test. The problem comes when you don't reject it. If you fail to reject that null hypothesis, is it because we really should fail to reject it, like up here, or is it because we lack the power to detect that difference? All right, so that's our relationship, and we'll talk about it again as I introduce these non-parametric tests. And then the other thing are these factors that influence our power. So the difference between the means that we're testing, the variation in our population, so your standard deviation, standard errors, and then your sample size. So if we're trying to detect differences in the means, and we have means that are close together, our test is going to have less power to detect that difference than if our means are really far apart. That should make sense, right? So the more different our means are, the higher the power of our test. Same thing goes with variation in our population. So if this is our means, all right, those are our two means, and we're trying to detect the difference between these values, we can have a small amount of variation. All right, that's going to give us high power. Or we could have a large amount of variation, and that's going to be a lower power. Our means are different. I try to draw the means that they're about the same separation. But in this case, we've increased the amount of variation that we have in each group, and that hurts our power. It makes it harder for us to detect a difference in our means. And the last thing is the sample size. So the sample size is related to these, to our variation, I should say our calculation of the standard error. So standard error is the standard deviation over the square root of our sample size. If we increase that sample size, we're going to cause an increase in power because we'll shrink our standard errors. And we use our standard errors to detect this difference. So things you need to know on this slide. The power. What affects the power? if we don't detect a difference in our mean, is it due to perhaps not having enough power to detect that difference? And if it is, how do we correct it? Well, we normally can't have any effect on the difference in means. You know, we can't tell a plant, hey, you need to grow bigger so I can detect the difference between you and this other population. That just doesn't work. A lot of times we can't control the variation unless it's measurement variation. Right? So that kind of leaves sample size. Boost your sample size. Maybe then you can detect the difference. I will say the one thing with the difference between the means, let's say you're measuring the effect of nutrients on plant growth, all right, and you did like 1x nutrients and then 0.5x nutrients, and you didn't really detect a difference in growth. You can look at it and say, yeah, the group with the higher nutrients grew a little bit taller, but it's not significant. Maybe one thing we can do is alter it. Instead of doing 1x and half x, let's do like a quarter or a tenth to try to increase that difference in between the means. That's a possibility as well. All right, now I present this because we do need to know what happens when we fail to reject it. Is it really, truly, the means aren't different, or did we lack the power? But also, you're going to get this question. People will ask, well, how much power do you have? You know, could you test it? Well, in order to know that, you have to have three things. You have to know the difference of the means. We have it. We can calculate. You also have to calculate the variance of the population. That's going to be a little bit harder, because how do we know what we actually calculated is representative of the population? And then we also have the sample size, which we do have. So you have to have these three things to calculate power, and yeah, a test will give that to us. R has a function that'll tell us the power of the test. Do we necessarily care about this? Not really. But what people do care about is this part, the sample size. And I get this. Students come in and say, how many samples do I need to run my experiment? Well, if you needed three things to the power, how many things do you think you need to determine your sample size? You needed three things to calculate the power. How many things do you think you need to calculate your sample size? Three things. We need the difference in the mean, the expected difference of the means. You need to know the expected amount of variation in your data set, and you have to specify the power. The problem that we have is that normally the students that come and ask me, how many do I need to do? They have no idea what the difference in the means will be or what the variance in the population will be. They didn't run the experiment yet. They don't know what they're going to expect. We don't know how tall those plants are going to grow. We don't know the variation of those plants. So coming up with a sample size, I can't do that. That's why I say do as many as you possibly can, as much as time and money will allow, so that we can get that. However, if you're doing a pilot study, that's a little bit different. The pilot study is aimed at getting difference of the means and the variance in the populations. Now, pilot study is a smaller type of study, uses smaller sample sizes because they're not really aiming to detect differences. What they're doing is trying to get this stuff, this information, so that they can then say, okay, based on this pilot study, I need a minimum of 1,000 individuals to take part in my survey. And if I have 1,000 individuals, then I'll have enough power to detect the difference in opinions of, let's say, 2 on our Likert scale, on that 0 to 5 scale. All right? So there is things that I would appreciate if you remember when you finish this class, and then it's this. All right? Don't come ask me, hey, I'm getting ready to do this study. How many replicates do I need? Yeah. What's your mean? What's the variance? What power are you shooting for? Most of the time we don't know that. But if you do a pilot study first and say, here's what I did on this short, 10 sample study, this is what I have. How many do I need to run to to get a significant p-value. Yeah, we could do that. We could do that. All right, so power's all about the ability to detect that difference when it exists. And things that we can do to influence that is sample sizes and try to take measurements that are as different as possible. Do we see this in real life? Yeah, what do we see? So I'll say, I laugh at a lot of studies that come out that make the news. They're sensationalist studies. Right? Yeah, right. Were you going to come in and ask me how what's the sample size?
No, actually, I just sat here and I realized that's part of the reason why if I could somehow manage to increase my sample size, which is why some of my genes wouldn't sequence.
Mm-hmm.
And so I have a limited number for the 6 genes that I'm looking at, but they're not all the same number. So like for one gene, I'll have like 16 samples, and for the other gene, I'll have like 6.
Yeah. It's concerning. But I guess the latest headline that I remember was COVID infections increase mortality rate across all causes of deaths.
What?
Right? That's what I thought. And then I'm sure, I didn't look it up, but I'm almost guaranteed that if you go look at it, you're going to have like 30,000 plus individuals in that study, which means like a 0.1% difference is going to be significant. That's why when you hear, oh, butter's good for you, no, butter's bad for you, no, butter's good for you, no, butter's bad for you. When you look at those studies, they've got so many individuals in the population that you can detect very, very minute differences.
It's not even grammatically correct.
But, what's that?
That's not even the headline. That's not even grammatically
Yeah,
correct. Like,
you know, it's sensation. Whatever. But you've got so many samples, and where that kind of, is it statistically significant? Yes, it is. Is it biologically significant? Probably not. Yeah, pay attention to that. Anytime you get a skeptical headline, we should check it out. So let's see here.
They do that with like beer and wine all the time. It's good for you.
Yes.
It's bad for you. It's good for you.
Yeah, wine is good for you.
Yeah.
Let's see if we can find. I think that came up. Let's see here. Wine decreases mortality, maybe. Here it is. Let's see here. I have to. It's harder. Oh. Medical news today.
Sounds like a trustworthy
Right.
source.
Lifestyle changes improve resistant high blood pressure, whatever that means. But I was looking for the red wine. 140 people. It's not bad. Wow. Yeah, let's see here. I thought they had it on here. Do a search. Wine. No, I don't know. They must have taken it off. The search had like 4 ounces of wine or something like that was showing a decrease. But again, a lot of that stuff you get such a large sample size that, yeah, you're going to detect the difference no matter what. So, all right. So that wraps up that portion. And what we are moving to are tests. All right. We're starting, we've introduced the idea of power, errors, everything. Now what we're going to do is move to our test. So we're going to do, this is a simple hypothesis test because it's one sample. All right, and what we're going to do is one sample continuous data. We're going to do our continuous data, and then we'll transition near the end to categorical data. So before we break for today, when we report the results, we have 5 items to report, 5 items to include. And this is in our write-up. So our exam had some of that where I said, report the mean and standard error as if you were reporting it in a paper. We're going to start having that all the time where it's write up your results as if you're reporting it in the paper. There's 5 things we have to report. One is the conclusion or the interpretation. This is different from saying we reject the hypothesis or we fail to reject it. Our conclusion or interpretation goes back to our biological question. So we start with a question, just a basic question. Are the means equal? Or are the means different? That's a basic question. And then our statistical question is the null hypothesis and the alternate hypothesis. This conclusion interpretation goes back to our basic question. All right, so we have to answer our question. We have to make that interpretation. We then have to state what statistical test was performed. We then have to give a p-value. We have to give our test statistic, and we have to give our degrees of freedom. Now, I listed test statistic and degrees of freedom as being optional because there are some tests that do not have a test statistic. There are some that do not have a degrees of freedom. All right, for example, our randomization test. We don't have a test statistic. We don't have a degrees of freedom. Can we report a test statistic that we collected from our data? Yeah, we could, but it's not required because our test didn't make assumptions about our data, so we didn't have a degree of freedom associated with it, and we didn't have a test statistic that we then compared to some null distribution. All right, so there's 5 things that we have to report: conclusion or interpretation, statistical test, p-value, test statistic, and degrees of freedom. When we report our p-value, we have to round it appropriately. Now, you've probably seen p-values written out completely, 5, 6 decimal places. We don't have to do that. If our p-value is greater than 0.05, we go to the hundredths place. So we go to the 0.01. That's what we do. If we are between 0.01 and 0.05, we go to 3 decimal places. And the reason we do that is if we had 0.044, right, that stays at 0.04, but if we have 0.046, that would round up to 0.05. And we just want to be clear to the reader that we didn't actually get 0.05. That's the rounded value. Our p-value is actually less than that 0.05. So anytime we have a p-value between these 2 numbers, we go to the thousandth place, 0.0001. If we are between that 0.001 and 0.0001, then we go to 4 decimal places. All right? And if we have any value less than this 0.0001, we just say p is less than 0.0001. We don't have to go any smaller than that. Our probabilities are significant. They're small. They're tiny. All right? So what we'll do is we'll just kind of refresh this, and then we will get right to our tests of means. And these are 1 sample tests. And what we're going to do, I'm working on this separate piece of paper as part of the accessibility stuff. I think what we're going to do is I'm going to post a document where every time we have a test, after we introduce it, you fill out the sheet that lists the test name, test statistic, hypotheses. You'll have to find a data set to use as like an example on how to do the test and have an example write-up and stuff. And that way, if you fill it out, or if you complete it and you print it out, you have a collection of all your tests summarized. So we can kind of do it as we're working through the class. All right? We'll take a break. We'll come back up for lab. We'll introduce our 1 sample tests, which are easy. You've already seen the z-test, believe it or not. I'll pass back those exams as well. So I am on this our markdown stuff. We just finished this, so I'm going to save it. With our 1 sample test, we're going to start a second R markdown. So that one's good.